All right, so I quickly made you a short little video to help you with question 11 and 12 because it seems to be a problem. If we look at question 11 and 12, it actually says there, okay, we have to actually start with this blue strand, which is called an mRNA strand. And it says that on this mRNA strand, all right, you need to, at question 10, tell us what the DNA looked like that made this mRNA strand. And then in question 11 and 12, tell me what happens afterwards. Okay, so this strand actually comes from the first page, which is this diagram. So this mRNA strand is the same one as the question is talking about. All right. Now, to figure out where this mRNA strand comes from, I actually just wrote down this mRNA. I copied it and I wrote it here. So that mRNA strand is what's given. Now, obviously, in order to make an mRNA strand like we've got here, we needed a DNA originally. All right. Now, remember, mRNAs will have U's. That's why the strand has U's right through. But don't forget, DNA will actually have T's, no U's. All right. So you have to think back now. What did the original DNA strand look like to make this mRNA? So that's not too complicated. The U would be an A. The A needed a T. So if the DNA strand had an A, it would attract a U. If the DNA strand had a T, it would attract an A. You would have needed a C to attract a G. So in that way, you can actually figure out what the original DNA looked like. G, G. Okay, so you needed DNA. You needed DNA to make RNA. Okay, so this mRNA was made in the nucleus. So all of this happened in the nucleus. This mRNA strand then came loose. It became it detached. It moved out of the nucleus and it went to a ribosome. So there we go. Now we're looking at a ribosome. Okay, so this mRNA strand is actually going to go and stick to a ribosome. As soon as it does that, what's going to happen is that you find molecules looking like this, tRNAs, okay, that float around in the cytoplasm. So I've made 3 here quickly, my art skills. Okay, so you've got 3 of them. They're floating around. They come from your diet. Whatever you've eaten, that's where they come from. Now you'll notice this thing is called a tRNA molecule. Now tRNA molecule, at the one end, it's got 3 bases, and the other end, it has the ability to attract an amino acid. Okay, and the reason why we call it tRNAs is because this tRNA, the T stands for transfer. What it will be able to do is that it will transfer an amino acid. The amino acid's floating around. The tRNA will pick it up, and the whole thing will be transferred to this mRNA strand. Okay, now you'll see that I've written down the bases on the mRNA strand in teams of 3. Each of these triplets on the mRNA strand is called a codon. Okay, and then if we look at the tRNA, there's 3 bases there as well. These 3 bases are called an anticodon. Now, in your question, your worksheet, it actually gives you the first tRNA. You can see that the codon in blue on the mRNA is AUG, which means the anticodon that corresponds to that will be UAC. That's why this tRNA with this anticodon is able to bond to this mRNA, because the anticodon sequence and the codon sequence, they match. So UAC. So question 11 actually says the first anticodon is given as UAC. Write down the subsequent ones. So if the first codon is UAC, very nice, they gave it to you. There's the UAC. That one will bond there, UAC, bringing with it a specific amino acid. Now question 11 asks, what will the sequence of these tRNAs be? So if the first one is UAC. Okay, somewhere I made a mistake. I don't know where I made the mistake. Oops. That should be an A. That should be a U. Sorry. Okay, that makes sense. Okay, so the next codon is UGC. So the anticodon that it will be able to attract would be ACG. Okay, and with it, the tRNA is going to bring an amino acid like that. Okay, the third codon is AAG. So that one will attract an UUC. So the A and the U, the A and the U, the G and the C. So in this way, we know that the third one over there, UCC, is going to be AGG. The fifth one is going to be CCU. Then we're going to have GUC, and that's it. So at question 11, that's the answer for question 11. The tRNAs, they give you the first one, UAC. The rest for 5 marks will be ACG, UUC, AGG, CCU, GUC. We know that because these anticodons bond in correspondence to the bases on the mRNA. So the mRNA strands bases in sets of 3 are called codons. They will attract specific tRNAs which has an anticodon end. So there's a tRNA again. The one side's got an anticodon. So that's how we figure out question 11. Okay, but then it says at question 12 that you need to use the key to write down the amino acid sequence, because what will happen is every time a tRNA bonds, it brings with it an amino acid. But the type of amino acid actually depends on the type of anticodon. As soon as these guys are adjacent, they will join. This tRNA can come off, okay, and be used later on again. This one can come off and be used later on again. This one will come off and be used later again. So in this way, this chain of amino acids become longer and longer. The moment we've got 50 or more amino acids, we've got a protein. But the order of these amino acids is determined by the order of the bases in the mRNA strand. So how the heck do we figure out which amino acid goes where? That's what we need the key for. So this key is super complicated. You don't need to know this off by heart. It'll always be given to you. So if we have a look, this key gives you the tRNAs. So let's look at this. What was our first tRNA? Now I have lost it. Our first tRNA was UAC. So on this circle, what we do is we start from the inside. The first base there was a U, so I'm going to go there, U. The second one is an A, then I'm going to go A. The third one is a C, I'm going to go C, which means UAC, and the amino acid is TYR. It stands for tyrosine, but you don't have to know that. You just need to write down TYR. Okay, the second tRNA we had there was ACG. So I'm going to do the same thing. I'm going to go to my key and I'm going to find the A on the very inside. There's the A. The next one is a C. So then in this quadrant, I look for the C, which is there. Then I go to the G, which is there, ACG, and it's going to be THR. So THR. So you're going to use the tRNA anticodons every time, all right, to take the key starting from the inside. First blocky, second blocky, third blocky, to read off the key.